Before trees ruled the land, they ruled the ocean. Brown algae, ancient, overlooked, and quietly essential to almost every breath you take. Yes, these are brown algae. In this video we will learn about seaweeds. What is seaweed? General structure, cultivation of Undaria, life history of Undaria, grow out of plant body, harvesting, marketing. We will also learn about structure, reproductive cycle and cultivation of laminaria. Before learning about cultivation of laminaria, it is necessary to know that what is seaweed. Seaweed is the common name for countless species of marine plants and algae that grow in the ocean as well as in rivers, lakes, and other water bodies. These are commonly known as marine macro and microalgae. They are plant-like organisms that generally live attached to rock or other hard substrata in coastal areas. Its structure comprises of holdfasts that act as roots to anchor the plant in the ground. Its upper part consists of a leaf-like structure known as blade and a central axis known as stipes that contain gas bladder. These gas bladders are known as pneumatocytes. Following are the main groups of seaweeds cultivated for human food. First one is red algae known as Rhodophyta. Its common species are Porphyra spp and Gracilaria spp, while Undaria pinnatifida and Laminaria are commonly cultivated species of brown algae, Phaeophyta. Enteromorpha compressa and Monostroma spp are common species of green algae. Seaweeds are mostly marine. Species differ on the basis of salinity and temperature tolerance. Edible seaweeds require lower temperatures, between 10 and 20 degrees Celsius, for rapid growth. Reproduction may be sexual or asexual. Some species of red algae exhibit a biphasic, gametophyte, carposporophyte type of alternation of generation, while others show triphasic, gametophyte, carposporophyte, tetrasporophyte. Another common method among most of the cultivated species is vegetative propagation. Let's see how brown algae can be cultivated. Its common example is Undaria, which is commonly known as wakme. This species is cultivated in many countries like Japan, Korea and China. A brown seaweed. Thallus fixed by a ramified holdfast with numerous haptera, the origin of a flat stipe with denticulated margins in young individuals, frond blade-like, lanceolated, extending from the tip of the plant for half to 3/4 the length of the plant and reaching an overall length to about 60 centimeters or even up to 1 meter. It has a midrib with undulating wing-like pinnate blades at the base. The sporophyte is golden-brown in color, with a lighter colored stipe. Undaria pinnatifida or wakme is a large brown kelp with a branched holdfast giving rise to a stipe. Just above the holdfast, the stipe has very wavy edges, giving it a corrugated appearance. The stipe gives rise to a blade that is broad, flattened and lanceolate. It has a distinct midrib. The margins of the blade are wavy. Plants can reach an overall length of 1 to 3 meters. Consumption of the macroalgae as a seafood is divided into categories. The processed midribs are consumed inside China, while the sporophyll and blade are mainly exported to Japan and other Asian countries. It contains valuable pharmacological compounds such as large quantities of protein, polysaccharide and fucoxanthin. It is a carotenoid, antioxidant activity, good for liver health, glucose management, and obesity. Fucoxanthin rich in various kinds of macro and trace elements. Therefore, it has several biological activities like prevention of hyperglycemia, suppression of chemically induced mammary tumors, anti-hypertension and anti-obesity. U. pinnatifida is found in low intertidal zones up to depth of 18 meter, but is mainly found at depth between 1 to 3 meters. It is able to grow on any hard surface to which it can attach, such as rocky reefs, mudstones, cobbles or even shells of abalone or bivalves. It often forms a thick, dense canopy that shades the sea life beneath it and exhibits high tolerance to sunlight and wave exposure. However, it is less likely to be found in areas of high level of freshwater influxes such as estuaries or mouths of rivers, owing to its preference for salinity levels of 30 to 33 ppt. It is an annual species with 2 separate life stages. The life history of Undaria includes an alternation of sexual and asexual forms. Asexual form is sporophyte, which is a macroscopic stage, usually present through the late winter to early summer months, while gametophyte is sexual form that is known as microscopic stage, present during the colder months. Pinnatifida favors cold water with a preference for temperature less than 12 degrees Celsius. In Asia, the native habitat of Pinnatifida, sporophytes grow quickly between 5 and 13 degrees Celsius during winter and spring. The growth of Pinnatifida sporophytes occurs in the meristematic zone between the stipe and blade, while its apical parts simultaneously degenerate. Its suitable temperature for growth is 10 to 15 degrees centigrade, while at temperatures above 20 to 23 degrees centigrades, sporophytes begin to degrade and die off, respectively. The optimal temperature for the growth and maturation of Pinnatifida gametophytes is about 15 to 20 degrees centigrades, and optimal growth is positively correlated with an irradiance of 10 to 80 micromole. In Asia, the release of zoospores occurs in water temperature between 17 to 20 degrees centigrades, with germination taking place at approximately 20 degrees centigrades. However, when water temperature is over 20 degrees centigrades, longevity of zoospores decreases and less germination takes place. This figure shows the life cycles of Undaria pinnatifida, which show alternation of generation. During summer, gametophytes, sexual plant develop by the end of the season, around September. The sperms are released which fertilize the ova within the oogonoeme to form the zygote. Develops into the sporophyte, asexual. Culture practices include seeding, growing phase, harvesting, and marketing. The systems of culture are essentially the same as for Porphyra. It includes seeding twine, seeding, mature sporophyll, twine frames, grow out of thalli, cultivation ropes, harvesting, marketing. Parental plants that are used for yearly spoiling production come from a farmed population. Parental plants are usually reserved on loan lines until end of June after commercial harvest at the end of April. The plants are cultured under relatively deep water to elongate the time at sea until seeding process, hatchery, starts. The spores are collected and reared on so-called seeding twines. Seeding performed in large concrete or plastic tanks, kept in cool shaded positions. Mature sporophylls, sporophyte plants, become partially dried, then placed in the tanks filled with fresh seawater. Other method is use of twine frames, which are arranged in layers inside the tank to collect the zoospores released from the sporophylls. After about 2 hours, they are transferred to large culture tanks with a depth of about 1 meter, in which the frames are hung vertically. The gametophytes and the young sporophytes develop on the strings throughout summer. The hatchery starts in June when seeding spore onto collector is performed and ends in late of September. Light intensity. The light intensity in the tanks is regulated to facilitate rapid growth, high survival rates. If the water quality deteriorates, it is exchanged. Fertilizers may be added if the growth is poor. Areas with short periods of seed rearing and a lower likelihood of fouling include tank culture of gametophytes can be avoided. But most seaweed growers find it beneficial to perform nursery rearing in indoor tanks. Grow out of thalli. Starts in autumn, between September and November, when the water temperature falls below 20 degrees centigrade. There is less likelihood of fouling by epiphytic organisms. An epiphyte is an organism that grows on the surface of a plant and derives its moisture and nutrients from the air, rain, water, in marine environments, or from debris accumulating around it. Twines containing the seed are set out on cultivation ropes in water depths up to 5 meters, depending on local conditions. These cultivation ropes are made of synthetic fibers, maybe 10 to 20 millimeters in diameter. Seed twines are attached to them at about 15 centimeters interval. In exposed areas, the cultivation rope is set out with heavy anchors, a sufficient number of floats. This system has made it possible to culture Undaria in deeper waters exposed to heavy seas. While in protected areas like bays, the ropes can be stretched out on rafts. Cultivation of U. Pinnatifida starts at sea by laying the long line horizontally and inserting spoling strings at intervals of 35 to 45 centimeters. These strings will hang in the surface water, usually within 1 meter of the surface. The interval between the horizontal long lines is 2 meters. Each individual long line is 8 meters in length. The initial spoling is roughly 1 centimeter long. They will reach to 2-3 meters between outplanting in October and harvesting in April. Each individual long line can produce approximately 80 to 130 kg fresh Undaria biomass. The main variables to final production weight are the location, water current and water depth where they are farmed. Undaria grows rapidly and become ready for harvesting after about 3 months. Some factors severely affect its growth, like heavy seas may damage the floating rope system. They aid rapid growth of the plants. Water temperature is a major factor affecting growth. After maturation, harvesting is required. The harvesting length of the talli is greater than 50 centimeters, done according to the growth pattern of the talli, which is itself very much dependent on the length of the growing period. Harvesting starts in February and ends in late April. Plants are harvested manually by cutting the plants off the main cultivation rope. Sporophylls and talli are packed separately and transported to the processing facility on the same day. Harvesting when there is dense growth and the growing period is long, the well-developed talli are harvested first. This helps the remaining ones to grow faster and enables repeated harvesting. Talli are harvested by cutting the upper portion first, allowing the remaining portion to grow. All the talli may be harvested at one time, when they have grown to the maximum size. Marketing sporophylls are frozen immediately, while the remaining parts are bathed in hot water for approximately 20 to 60 seconds. The cooked plants are then thoroughly salted. The salted plants are packed in bags and pressed with heavy objects overnight to remove the excess salt water. Thereafter, the midribs and blades are separated manually by hand, packed into boxes and stored at low temperature. Thanks for watching. Like and share this video. Subscribe this channel to get more informative videos.